Hello and welcome to the Out of the Lab podcast, where we discuss the operational challenges in cell and gene therapy. Each episode, we sit down with a different voice in the space, from executives and scientists to patients and advocates, to figure out exactly where the industry is breaking down and how to fix it. Today, I'm joined with Tatyana, the head of cGMP operations at Harvard. Tatyana, welcome to the show.
Thank you. Thanks so much for having me. It's a pleasure.
Well, today, Tatyana, we are going to discuss one very interesting topic, which is the cultural divide between GMP operations and research environments. So to sort of start off, you have an academic background and you currently are in GMP operations. I'd love to hear your perspective on where does this come from? Why do people look at things in different ways in these two different contexts?
Yeah, and the difference in context turns out is surprising, right? So we are at a strange intersection here. We are in an academic setting, and we certainly have an academic research lab arm, but we also have a GMP component. And so as somebody whose background is entirely academic, I know all too well that nothing GMP, not in terms of language, not in terms of setup, not in terms of documentation expectations, to say the least, bears any resemblance to the research environment. And what happens is, especially nowadays, more and more therapies come from labs, right? They are the product of multiple years, decades of lab work and collaboration. And so the end product, which is what ultimately enters an IND or an FDA submission, is largely derived from a very long history of lab work carried out with the lab ethos, right? And so all of a sudden you have this wonderful new therapy or product and you're ready to begin an early phase small scale clinical trial. So in the case of cell and gene therapies, that would be a very small scale initial production with a small number of patients, but you need a GMP environment to manufacture your product. And so all of a sudden you have this need to transfer all of that knowledge, all those protocols derived in the lab into a GMP setting. And more than that, you want to make sure that your people understand that a GMP room, right, a clean room, is not what GMP is all about. There's so much more around it, right? And it has to do with a full-blown documentation infrastructure that enables traceability and deviations and data integrity and so on and so forth. And now we're really talking about something that's very concrete and specific, so that if you imagine somebody who's never worked on this process before, just for the sake of a hypothetical, when they read the GMP level instructions on how to manufacture the product, they can replicate it, they can do it, right? And the cultural divide, which is a perfect name for it, I think really comes from the fact that up until having to enter a clean room or having to make a product that is transferable to a GMP setting, a scientist often would not have ever encountered GMP requirements. And even if they have in some capacity, more than likely, they will not have been exposed to the full-blown scope of what that entails. And particularly in terms of documentation. This is really where things become quite burdensome for people who are on the development side. And so what happens is you end up on two opposite sides of the table with a very talented research team who's done a lot of great work. They've prepared a product that is very promising, and it can be tested, but they have done it a certain way. And the requirements in a research lab are different from the requirements in a GMP. And all of a sudden, you need to perform some kind of tech transfer. In other words, you need to transfer their process into a GMP environment. And it turns out that while the process is reasonably well described, perhaps, it's nowhere near in sufficient detail required for compliance, right? So all of a sudden, you go to this team of people who've been developing this therapy for a decade, and you're telling them that, in a sense, right, it's not quite up to par, that it needs to be written out a lot more specifically, a lot more concretely, things that are perhaps implied or not quite stated as hard facts must be very explicit. And it's just the level of description you need to get into is quite a bit more elaborate for GMP. And so it's difficult. That conversation is usually one that is not easy to have. And in my experience, it's often the case that scientists who don't understand that this is a requirement from a regulatory perspective, right? They may misinterpret this insistence on better documentation, better detail, more explicit wording of processes as kind of a strange demand on something that they have seen already work quite well, and they probably take a lot of pride in, and rightfully so. And so I found that it actually helped at one point to get people really just to see the way the regulation is written, to get really specific about, here is why we ask you to do this, and here is what it means that you are part of manufacturing, and here is the scope of that term definition on part of the FDA. And once you start speaking in those terms, then we really begin to have a dialogue. Then scientists don't have experience in GMP realize that this is not just some imposition on them, this is just what we're facing. And then it becomes more plausible to them that they could accept that new challenge. But even so, the hardship continues, and it will take multiple iterations for a single process to be truly ready for being brought into a clean room.
Yeah, I mean, that makes so much sense. And the first thing that comes to mind is really the purpose of the documentation. So when we look at, a research paper, generally speaking, its purpose is for another scientist somewhere else to continue building or doing something else with that research. And when that happens, it doesn't necessarily have to work 9 or 10 out of 10 times, because the stakes are generally not that high. It's the worst case scenario, someone wasted maybe a day or something like that. But when we're talking about GMP manufacturing, the stakes are different and the purpose of the documentation is different. So yeah, I would love to hear your sort of thoughts on that part of it.
That's a funny thing to say. That's an interesting observation, right? So when you are in a research lab and you perform a set of experiments, sometimes very elaborate, sometimes less elaborate, the idea is that reproducibility should at least be part of the equation. And there is a good faith and also an evidence-based understanding that the methods of the paper should describe in sufficient detail the processes that were carried out and the analyses to at least enable somebody to rebuild that experiment somewhere else. Now, that's fair enough, and absolutely, I think all of us would agree that reproducibility is absolutely critical. So the better job, a team does in really describing with great fidelity their methods, the more likely it is that reproducibility will be a fact. But at the same time, and you get to share raw data and all that stuff. But there is always the possibility that you have some variation between two different sites or even two different teams in the same site, and that is accepted very easily as common sense. And as you said, rightfully, the stakes are very, very different, right? And if you consider the converse of that, right, in cell therapy, whether that be, autologous cell therapies or allogeneic therapies, whatever it is, you are either essentially saying my product will always be the same, or in the case of autologous therapies, you really are saying this is the process, and that process will always be the same between batches, right?
Mm-hmm.
And so the purpose of reconstructing the steps to production is quite a bit different from the purpose of really describing your methods in a research setting. So that's important to understand. And the purpose is not a singular thing, right? So you want to ensure traceability in great detail, date, time, hour, who did what, when exactly, in as much detail as possible. You also want to make sure that that very same process is repeated with as much detail as possible in the same way, every time. So you have some uniformity.
Mm-hmm.
You also want to be able to conduct kind of a broader exploration in case retroactively, perhaps, you need to understand better an adverse event or some sort of incident or deviation, or perhaps, God forbid, trace contamination. Anything that is not super standard becomes the subject of investigation, and that's for very good reason, right? You need to be able to get to the bottom, to the root cause of hopefully any problem you encounter, and not just get to that root cause. You need to be able to describe the process by which you did that. Then you need to address that root cause with some kind of plan or action that would correct it or prevent it in the future, and so on. And it's not enough to have taken that corrective or preventative action and call it, right? You need to then monitor and demonstrate that, in fact, that action did achieve what you intended it to. Because if it didn't, not only are you in trouble in terms of safety and quality, but it also means that the method used to identify the root cause was possibly flawed. And so then you need to go back and reconstruct as much of the environment on that given day as is humanly possible. So that's one big part of why documentation is so critical. Another big part, aside from traceability, is that you need to be able to show down to the identity of ingredients, what is it that you did step by little step, right? So you wouldn't just say, well, we went in and we changed media. That would be absurdly vague, right? You cannot be doing that. And so there are teams of two. There is someone who is a verifier who then walks the operator through different steps. Those steps are, given as instructions in great detail. The full identity and lots of all ingredients are well described, so are materials. The flow of personnel to prevent contamination, to ascertain that indeed the environment was safe enough to perform a process. All of these are absolutely necessary, and they're necessary for a good reason. Not only is it for the safety and integrity of the product, but ultimately all of this is to protect the patient, right? And I will say it's enormously costly in all kinds of ways to omit any of these steps in GMP. And that isn't true in research, certainly not to the same extent, right? So if you are not thorough in a clinical environment, and it turns out that you notice halfway through your production process that something's gone wrong, whatever that is, you need to quit. You need to start over. You need to first analyze your process, analyze what people were doing, analyze your training. You need to perform a very comprehensive analysis of what went wrong with the hope of identifying what went wrong. But that just means your operations have now come to a stop. Whatever you've made to that point likely not usable. So what does that mean? That means that you need to start from scratch once you're ready to begin. That means that your materials need to be completely replaced, that your product needs to be remanufactured. That means that there is that much more time left between product and treatment, right? Administration to the patient. So not only is it expensive financially, as it of course is, but you also lose a lot of time. And most importantly, you really waste time in terms of getting the patient what they need, right? So the scale of what it costs to not be able to have really good control of your processes is massive. In research, while certainly it is costly to make mistakes, it is costly to not be able to recover the plan of action. I think we can all agree that the stakes are indeed lower, right? There isn't a human patient on the line. There isn't a production timeline. There is, and cell and gene therapies are exorbitantly expensive, right? So anything you add by having an imperfect process is on top of that. And so it compounds an already high cost of production. So you're absolutely right. The rationale for having documentation in GMP is very different. The purpose of that documentation is very different from that in research. And in research, you're often taught that you need to document, you capture the process, you capture what you're doing, whether that be in notes and whatever. And certainly, some key components, dosing, whatever, concentrations. But, the difference in vagueness in language is quite substantial. And I think it would be very familiar to many when I say many of us scientists are we viscerally understand what our process requires. But visceral doesn't count. I have a good friend who says nice doesn't count. It's like that. Visceral doesn't count. You need to be able to articulate it. More than that, you need to be able to justify it once it's put into human linguistic terms. And you need to make it concrete, because now this is what we're going to train our people on. And this is what we're going to use for subsequent batches. And that's really where trying to replicate a process becomes a very big deal. I was not long ago part of a conversation, and we were talking about validation runs, meaning process validation runs right before you start clinical manufacturing. And somebody said, well, tech transfer will be seamless because this person did 4 or 5 research runs and they all turned out very well. Let the alarm bells, right? And so the logical question is, that's fantastic. It actually is great, right? They've repeated their process in a research setting, and every time they get the same outcome.
Mm-hmm.
Excellent. That is fantastic. The first question that comes to mind is, so do they have batch production records? In other words, do you have GMP grade documentation for every single day of production? Crickets, right?
Okay,
and so tech transfer is not going to happen unless you have that.
Okay, the first thing I would love to sort of get into there is, for an audience that might be composed of some sort of founders in the cell and gene space approaching this, we're talking about the costs in terms of time to patient access as well as financial costs. Are we talking weeks, days, months? Like, give us some sort of frame, like how expensive can this be?
Massive expensive. Yeah. So of course, it depends on what kind of process you're working on. If your entire protocol is 3 days long, then your delay would be shorter than if your entire manufacturing process is 40 days long. But let's take the 40-day process. If at any point you have some kind of a deviation that really causes your product to not meet specifications, Or perhaps it causes your environment to not meet specifications. And so you can't really make your product because the environment is substandard. And you stop. In the case of many cell and gene therapies, the production is somewhat continuous, right? So you start with some material and then you continuously work on that material until it becomes whatever you want it to be.
Mm-hmm.
If at any point you must terminate, wherever that point is, you essentially scrap more often than not. Hopefully you don't, if somehow you have a process that can withstand that, but more often than not, you scrap whatever's happened up to that point. Then you must analyze all of your records of that process. You must analyze the environment. And again, who did what when. You have to figure out what exactly took place. Why did this happen?
Mm-hmm.
That doesn't happen in your head, right? You need to have a scientific, logical, coherent approach that could be written down as it should be, because that becomes part of your report on that deviation. And once you think you've identified the cause, it's time to set up your logical corrective process. And then you got to test it. So just this part will often take you easily a month. This is just reviewing your records, figuring out where things likely went wrong, explaining why you think that, explaining what the correction is, explaining why you think that's the correction, performing the correction, and that CAPA should stay open until you've really confirmed, continuously, repeatedly confirmed, that the correction was an appropriate call for the root cause. If you're unsuccessful in that, you can't start, because that means you don't have a root cause. So now we're talking, if it took you a month to figure this out, now we're talking, okay, let's go back to another month, right?
Mm-hmm.
If you then finally get to the point where you have your root cause, you have your corrective plan, you begin implementing it, now you need to go back to the drawing board and make sure that you have all of your starting materials, be that reagents, starting tissue, whatever. And you have to start from scratch to build up to where you left off so that you continue. So I often say, if you have a delay of a week, it's usually a 3 to 5 week delay, really.
Mm-hmm.
It is approximately, for small scale, early phase trials, it's often 3 to 5 times the time that
Mm-hmm.
you've lost. That's massive, right? In any way you measure it. It's massive in terms of, wasted income, wasted materials, recovery of materials, receipt of materials, testing, entry into your clean room, getting, so it is very, very substantial. It doesn't translate to one-to-one, you lose this is how much the cost truly is. It's exponential. It's not linear.
Yeah.
And so it will cost you quite a bit, especially because also in clean rooms, let's not forget that there are scheduled things you need to do for your facility. So if that falls somewhere in that period, that increases the amount of time you should be waiting until you can use your facility again safely. So sometimes it can be months, right? It's bad news.
Yeah, that makes a lot of sense. So what I'm really hearing is that these corrective and preventative action investigations and finding out what really was the thing that caused that initial deviation to start with, that is the sort of bulk of the expensive, this is your new science project, basically.
It really is.
As it starts. Yeah. And then aside from, that's a huge amount of personnel time. Also, these engineering runs that are happening probably each time that you're trying this, those can go well into the sort of 6-figure ranges just to run those. So it is clearly expensive.
Absolutely. It's very true. And that's why, it's a very good idea. And I'm sure most know this, that yes, you do have to have those so-called validation runs or engineering runs, but they're is occasionally a push to omit those runs
Mhm.
because of what people think is a robust tech transfer, thanks to the fact that a process has been successfully replicated in a research setting. And what people forget is it's one thing to replicate a process in the setting that you originally had it. But if you're moving to a new facility, you have different conditions. So your process validation somewhere else doesn't automatically transfer, right? And so the best, smartest things that that people can do is have the so-called, pre-validation dry runs, where you just figure out
Mhm.
what that new facility, new environment puts you in a position of doing that may be different from what you're used to. Because guess what? Being in a bodysuit that's completely sterile is not the same. And it's not the same feel as being in a lab coat or scrubs by safety cabinet. Those are very different things. So even those changes, let alone the changes in conditions, can contribute to changes in outcome. And so you need to validate your process for a new facility. But engineering runs, yes, super expensive. Dry runs before an engineering run in the long run will save money quite a bit. So, what we've been doing is we did a dry run, then we did a second dry run, then we did kind of we call it a sandbox run. So these are all just basically runs that uncover various vulnerabilities in your process. And then you quickly improve your process control, right? Update your documentation so that your new SOPs reflect those changes. And while that's a burden to do up front, imagine the burden it saves you when you're actually producing something for the clinic. It's incomparable. So yes, CAPA is expensive. Hopefully, if people have a good quality department, they can do it well. Engineering runs are terribly expensive. Delays in getting treatment to expensive on so many levels, especially if the patient needs to travel, that may get, or if they have to be seen by, I don't know, some kind of physician to be re-cleared as suitable for receiving the product, and so on. So it is a very diverse set of costs that come upon you and compound. And so if you don't, go back to tech transfer, if you don't have very, very quality documentation, very detailed, proper records, you're lost. And God forbid, the FDA comes and audits you. And like, oh, you report deviation. But as far as we can tell, this is a very inadequate analysis, and your CAPA maybe worked, maybe didn't. They pay attention to these things. And we all know the very unpleasant 483 letters that they publish on their own website. So this becomes public knowledge. It's not good.
Mm-hmm.
So investing in the right kind of documentation and documentation habits and process descriptions early on will save you a world of hurt.
Wow, it's been super insightful. And, the last thing that sort of comes to mind for me is I think it's important looking forward to remember that the whole goal here is regulatory approval. That is what all of this is for, is to get a stamp of approval so that you can sell your drug. So anyway.
That, and also you really want to be helping patients, right? So hopefully that's also part of
so
the
that
goal.
you can help
Yes,
patients.
regulatory approval is necessary so that you get your product to commercialization, so that yes, it can be prescribed, you can get it to patients. But the biggest deal is that you only get, that's the idea anyway. You get regulatory approval because you're doing things safely.
Mm.
If you're not doing things safely, and maybe you don't get an audit, and so it keeps going, sooner or later there will be an adverse event. And this is not what anybody should be about, right? Yes, you want to sell a product. Yes, you want to administer a therapy, but you want it to work and you want it to be safe. Mm-hmm. And you do that if you have a robust process.
Wow, it's been a super insightful conversation. Thank you so much for joining us on today's episode.
Yeah, thank you. My pleasure. It's been great to be here. Thank you.